For more than 2,000 years, historians told the story of ancient Persia the same way, as a story of conquest. The Achaemenids built the empire. Alexander's Greek armies tore it down. A tribe of horse-riding nomads from Central Asia rode in and seized what was left. Then a new Persian dynasty rose up and reclaimed the throne. 4 dramatically different ruling powers in roughly 6 centuries, and for generations, the working assumption among historians was straightforward. When the rulers changed this, dramatically, the people underneath them changed too. New conquerors, the thinking went, generally meant new populations settling in, intermarrying, and gradually replacing whoever came before. In 2025, a team of geneticists finally tested that assumption directly against buried human DNA rather than against palace records and battle accounts. And what they found underneath the entire narrative was something nobody expected: a single genetic thread that ran quietly through every single one of those regime changes, almost completely undisturbed. This is the kind of question that sounds simple until you actually try to answer it. Written history tells us who conquered whom and roughly when. It tells us almost nothing reliable about what happened to the millions of ordinary farmers, herders, and craftspeople who were already living on that land before the soldiers arrived, and who kept living there long after the fighting ended and the new rulers had settled into their palaces. For most of human history, those people didn't write anything down, and even when court chroniclers did bother to mention them, it was rarely with any interest in where their ancestors had actually come from. Genetics offered for the first time a way to ask that exact question directly, by reading it out of the bodies of the people themselves, rather than out of any text written about them. To understand why the finding matters so much, you first need to understand exactly how dramatic Persia's political history actually was. The Achaemenid Empire, founded by Cyrus the Great in the 6th century BCE, once stretched from the Balkans to the edge of India, the largest empire the ancient world had ever seen. Then, in 330 BCE, Alexander the Great's armies swept through and dismantled it, installing Greek-style Seleucid rule across most of the Iranian Plateau. That Hellenistic government didn't last forever either. By the middle of the 3rd century BCE, a nomadic Central Asian tribe called the Parni, sometimes referred to as the Aparni, rose up out of the steppe, overthrew Seleucid authority in the region, and established what would become the Parthian Empire, a genuinely foreign group of horse-riding outsiders suddenly running one of the most sophisticated civilizations on Earth. Centuries later, the Parthians themselves gave way to the Sassanid dynasty, which ruled Persia until the 7th century CE. On paper, this reads like 4 almost entirely different populations taking turns controlling the same territory. Each one of those transitions came with real, documented upheaval, not just a change of flags on a map. Alexander's campaign across the Persian heartland involved years of sustained warfare, the burning of the Achaemenid ceremonial capital at Persepolis, and the deliberate installation of Greek administrators and, in some regions, Greek settlers across the conquered territory. The steppe culture, its own distinct language, and its own military traditions built around mounted archery, suddenly sitting atop a settled agricultural civilization it had no prior connection to. Historians studying this period have long debated exactly how thoroughly each of these transitions reshaped the population beneath the new rulers, and without direct biological evidence, that debate had nowhere solid to land. A team of researchers led by geneticist Motahara Allah Amjadi, working with colleagues at Eötvös, Lorand University, and the Institute of Archaeogenomics in Budapest alongside Iranian researchers at the University of Tehran and the Iranian Center for Archaeological Research, set out to test whether the DNA buried in ancient Iranian graves actually matched that story of repeated population turnover. Publishing their results in the journal Scientific Reports in May 2025, the team analyzed 50 human samples pulled from 9 archaeological sites scattered across Iran, successfully sequencing 23 ancient mitochondrial genomes and 13 full nuclear genomes, spanning an almost unbelievable stretch of time, from roughly 4700 BCE all the way through 1300 CE. The methodology behind this kind of research is worth understanding in some detail, because ancient DNA doesn't come easily, especially from bones this old and this widely scattered. Researchers prioritize sampling the petrous bone, a dense structure at the base of the skull that happens to preserve genetic material far better than almost any other part of the human skeleton, along with teeth and long bones when petrous samples weren't available. Back in dedicated ancient DNA laboratories in Budapest, technicians extracted whatever fragmented genetic material had survived the intervening millennia, built sequencing libraries out of it, and used targeted enrichment techniques to fish out the specific genetic regions most useful for ancestry analysis. Radiocarbon, dating pinned down exactly when each individual had lived, and once the genetic data came back, the team ran it through several different types of population genetic analysis, comparing ancestry proportions, mapping out clusters of shared genetic drift, and modeling exactly which ancient reference populations best explained each individual's genome. None of this happens quickly or cheaply. Ancient DNA degrades over time into short, damaged fragments, often contaminated with genetic material from soil bacteria, fungi, or whoever handled the bones after excavation, sometimes centuries after the original burial. Getting usable results meant working in dedicated clean room facilities, specifically designed to prevent modern DNA from contaminating these ancient samples, screening every sample first with shallow sequencing to check how much genuine ancient human DNA had actually survived, and only pushing forward with the far more expensive deep sequencing on the handful of samples that cleared that initial bar. Of the 50 samples the team started with, only a fraction ultimately yielded high enough quality genetic data to support the kind of detailed ancestry modeling this study relied on. A reminder of just how much painstaking laboratory work sits behind even a single confirmed ancient genome. 3 specific sites anchored the historical portion of this study, each one corresponding to a different chapter of Persia's political history. Marsin Chal, in Iran's Semnan province, held burials from the Achaemenid to Seleucid transition, roughly 355 to 280 BCE, capturing individuals who lived right through the fall of the original Persian Empire and the arrival of Greek rule. Lear Sangbon, in Gilan province near the Caspian Sea, held burials from deep within the Parthian Empire itself, roughly 247 BCE to 224 CE individuals living under the rule of those same horse-riding steppe conquerors. And Vestama, in Mazandaran province, held burials spanning the transition from late Parthian rule into the Middle Sassanid period, roughly 200 to 460 CE. Lined up together, these 3 sites essentially formed a genetic checkpoint system, sampling the population at 3 separate moments spanning nearly 600 years and 3 completely different ruling dynasties. If watching researchers extract a genetic paper trail out of skeletons, this old, spanning literal empires rising and falling, is exactly the kind of ancient mystery you come here for, go ahead and hit subscribe, because untangling stories like this is exactly what this channel exists to do. Here's what the genetic data actually revealed once all 3 sites were compared against each other and against the wider dataset of previously published ancient Iranian genomes. The core ancestral signature running through the population of northern Iran, a genetic blend rooted in Iran's own early Neolithic farmers, combined with a component related to hunter-gatherers from the Caucasus, remained the dominant genetic thread across every single one of these historical periods. Individuals buried during the Achaemenid-Seleucid transition at Marsin Chal, individuals buried deep within Parthian territory at Lear Sangbon, and individuals buried during the shift into Sassanid rule at Vestama all showed the same underlying ancestral profile, with only modest regional variation layered on top. In the researchers' own analysis, most of these historical individuals could be genetically modeled using this single ancestral source without needing to invoke any large-scale replacement event to explain their DNA. That finding directly challenges the assumption baked into the standard historical narrative. The Parthians weren't a local dynasty. They were nomadic outsiders from the Central Asian steppe who violently seized control of an already ancient civilization. If conquest generally came bundled with substantial population replacement, the way it so often has elsewhere in history, you'd expect Parthian-era burials to show a meaningfully different genetic signature than Achaemenid-era burials that came before them. Instead, the researchers found that broader steppe-related ancestry, the kind of genetic signal you'd expect from a large scale, Parthian migration actually reshaping the general population, remained a distinctly minor presence in the historical samples. The rulers changed dramatically. The underlying population, at least in the specific communities this study sampled, largely didn't. This pattern held up across multiple independent lines of genetic evidence, not just the broad ancestry modeling. When researchers separately tracked maternal lineages, passed down through mitochondrial DNA, and paternal lineages, passed down through the Y chromosome, they found the same story of deep continuity. A Y chromosome haplogroup, J1, a lineage the researchers estimate originated in the Western Persian Plateau roughly 20,000 years ago, appears consistently from the Bronze Age, straight through into the historical Achaemenid to Sassanid samples. A related lineage, haplogroup J2, follows a similar pattern, rising in frequency during the Neolithic, peaking during the Bronze Age, and remaining a consistent presence all the way through the historical period. These aren't lineages that show up, vanish, and get replaced by new arrivals with each political upheaval. They're lineages that simply persisted, quietly, generation after generation, underneath a rotating cast of conquering dynasties who mostly left the actual gene pool of the people they ruled largely intact. Here's where the story gets genuinely more complicated, and where the hidden bloodline this study's title points toward actually comes into focus. Not every site told exactly the same story, and the exception turned out to be Lear Sangbon, the Parthian-era cemetery in Gilan province. While most individuals at Lear Sangbon fit the same continuous local ancestry pattern found everywhere else, the researchers found that modeling this population required something the other sites didn't, an additional injection of Western genetic ancestry, connected to populations from Anatolia, the South Caucasus, or the Eastern Mediterranean, making up somewhere between 16 and 26% of certain individuals' ancestry. This wasn't a subtle statistical footnote either. It showed up clearly across multiple different types of genetic testing the team ran, and it lined up with something archaeologists had already noticed independently at the same site: non-local artifacts of Egyptian origin, dating to the Roman era, recovered from the same burials. In other words, buried within an otherwise remarkably stable, continuous Persian population was a small cluster of individuals whose ancestry told a genuinely different story, one connecting them to trade networks and possibly direct ancestry stretching westward toward the Mediterranean world, hiding in plain sight within a cemetery that, on the surface, looked like every other Parthian-era burial ground in the region. Long before anyone could read DNA, the archaeologists digging at Lear Sangbon had already sensed something was different about this particular community, based purely on the foreign objects they found buried alongside the dead. The genetics simply confirmed with far more precision than any artifact ever could that these individuals really did carry ancestry that set them apart from their neighbors just a few hundred miles away. Lear Sangbon produced two other genuinely remarkable findings that round out this story. Using a statistical method built specifically for detecting biological relatedness in degraded ancient DNA, researchers discovered that two individuals buried at the site were, genetically speaking, essentially identical twins. That result wasn't just a laboratory curiosity either. Archaeologists had already recorded independently, and years earlier, anthropological evidence suggesting these two individuals were twins based purely on skeletal analysis and burial context. The DNA didn't just confirm an educated guess. It transformed a plausible archaeological hypothesis into hard genetic fact. And it's a genuinely rare thing for ancient DNA to be able to do: confirm the specific and this personal, a detail about two people who lived and died more than 2,000 years ago. The second finding from the same cemetery cuts in a very different direction. Among the individuals analyzed at Lear Sangbon, one showed an unusually high degree of what geneticists call runs of homozygosity, long unbroken stretches of identical genetic material inherited from both parents. A telltale sign that this particular individual's parents were closely related, likely first or second degree relatives, a parent and child, or siblings, or an equally close family pairing. That's a strikingly different genetic story from most of the other individuals sampled at the very same site and time period, several of whom showed almost no such signal at all, indicating they came from a much larger, more genetically diverse mating pool. Within a single Parthian-era community, in other words, the DNA reveals both a small foreign-descended lineage with distant Mediterranean roots, and separately, at least one case of close family inbreeding sitting right alongside individuals from an entirely unremarkable, broadly connected local population. Taken together, these findings paint a picture of Lear Sangbon as a genuinely unusual place, even by the standards of an empire built on trade routes and cultural exchange. Most of the people buried there lived and died as part of the same broad Iranian population that had occupied this region for thousands of years before them. But scattered among those ordinary burials were individuals whose lives told very different stories, twins raised and buried together, a family close enough in blood to leave an unmistakable genetic signature, and at least a few individuals whose ancestors had, at some point, traveled from far to the west and settled among people whose own roots ran in an entirely different direction. A single cemetery, in other words, managed to preserve evidence of nearly every kind of family and community structure a population geneticist could hope to find, all within a few hundred burials in one. It's worth being precise about what this study does and does not claim, because a finding like this can easily get flattened into something more dramatic than the underlying science actually supports. This is not a discovery that some entire ruling bloodline running through the Persian throne itself was secretly fraudulent or illegitimate. The researchers never sampled the actual Achaemenid, Parthian, or Sassanid royal family directly, and nothing in this study makes any claim about the biological legitimacy of any specific king or dynasty. What the data does show is that the broader population living under these successive empires remained far more genetically stable than the standard historical narrative of repeated conquest and replacement would predict, with one clearly documented, genetically distinct lineage embedded within a single community at Lear Sangbon, standing out as the meaningful exception to that broader pattern of continuity. The researchers, themselves were also careful to flag the limitations of a dataset this size. 50 samples across 9 sites with only a handful of individuals representing each specific historical period is a meaningful start. But it's a small window into what was, at its height, an empire home to tens of millions of people spread across a territory the size of the continental United States. The study focused specifically on northern Iran, meaning its conclusions about population continuity may not automatically extend to other regions of the empire, particularly the southwestern heartland where the Achaemenid capitals themselves were located, an area the researchers explicitly noted as underrepresented in the current ancient DNA record. Broader claims about the genetic history of the empire as a whole will require considerably more sampling from a wider geographic spread, including the empire's southern and eastern provinces, before the picture is anywhere close to complete. There's also a valuable methodological lesson buried inside this study, separate from anything specifically about Persia. For decades, the historical record we have access to was written almost entirely from the perspective of rulers, court chroniclers, and the winners of military conflicts, a record that naturally emphasizes dramatic ruptures, invasions, and the replacement of one ruling elite by another. Ancient DNA offers a fundamentally different kind of record, one written not by scribes with political interests, but by biology itself, and biology, it turns out, often tells a much quieter, more continuous story than the history books do. Dynasties rise and fall in the historical record in dramatic, clearly demarcated chapters. Populations, this study suggests, tend to persist through those chapters far more stubbornly than the chapter breaks themselves would ever suggest. Step back far enough, and this becomes a story about the difference between political history and human history. Two things that get treated as identical far more often than they should be. Persia's throne changed hands violently and repeatedly across the centuries. This study examined from native Achaemenid kings to Greek-descended Seleucid governors to Parthian steppe warriors to the Sassanid dynasty that would eventually give way to the Islamic conquests. Yet underneath every one of those transitions, in the specific communities this research sampled, an older genetic thread kept running largely undisturbed, occasionally joined by small pockets of newcomers like the individuals at Lear Sangbon, but never fundamentally replaced. The bloodline that mattered most to the actual population of ancient Persia wasn't the one sitting on the throne. It was the much older, much quieter one running beneath it, invisible in any surviving text, and only now, through the careful reading of ancient bone, finally becoming visible at all. There's a reason this kind of finding tends to surprise people more than it probably should. We're used to thinking about history through the lens of maps that suddenly change color, empires that appear and vanish in a single textbook paragraph, dynasties that feel like entirely separate chapters in a story. Genetics doesn't read history that way at all. A genome doesn't know or care which king happens to be sitting on the throne in a given decade. It simply keeps recording, quietly and continuously, the accumulated history of who had children with whom, going back generation after generation, indifferent to whatever political drama happened to be unfolding above it. That mismatch between the sharp, dramatic chapters of political history and the slow, continuous record written in DNA is exactly what makes ancient genomics such a valuable, and occasionally humbling, corrective to the stories we thought we already understood. If a story like this, watching researchers pull a genuinely different version of history directly out of buried DNA, is exactly the kind of deep dive you come here for, make sure you're subscribed, because there's a lot more ancient history waiting to be quietly rewritten. This way. And if this particular finding surprised you as much as it clearly surprised the researchers who dug it up, let me know in the comments which other ancient empire you'd want tested against the hard evidence next, and I'll see you in the next one.